Okay, welcome to A-Level Biology, the maths. This is part two at Let's Get to the Marks. Let's go through some A-Level Biology maths questions. So here we have three graphs to do with the heart. Figure 3. We've got the pressure in the left ventricle in kilopascals. We've got the pressure in the aorta near the heart in kilopascals, and we've got the rate of blood flow in the aorta near the heart. And what you've got to do here is use information from figure 3 to calculate the heart rate of this dog. So pause the video and have a go. Okay, right. I'm using a highlighter just to identify some of the key points. Like we need it in beats per minute, not beats per second. But the time we've got at the bottom of all three graphs is seconds. And all you have to do is find the same point on each graph on the curve and then go to the next identical point on the curve. So we could do it from P to P in the top graph or Q to Q. I'm going to find 2 points that are easiest for me to do the maths deduction. So here I'm finding it easy at 0.2. I'm going to go to that little dip in the graph there. Then I'm going to find the identical dip further along in the graph, and that's over here, what looks like 0.56, and that tells me that's one whole cycle of the heart, and so that's 0.2 to 0.56. And if I do 0.56 minus 0.2, I'm going to get 0.36, and that is the time for one heartbeat. Now I need to find out how many heartbeats are in a minute. So it takes 0.3 seconds for one beat. So how many beats will be in 1 minute? Well, I then need to do 60 divided by 0.36, and I'll find out the beats per minute. And there we go. So don't forget to like, share, and subscribe. Really helps with the channel. Helps me get the videos out there to people so they can do well in their A levels. And yeah, going to move on to the next question. So here the answer is going to be 166, which is quite high, but dogs have a lot higher heart rates than humans. Being smaller animals, you'll find if you went down to a mouse, it would have a much higher heart rate than this even. Okay, let's move on to the next question. Okay, for this next question, when making up extraction solvent E, the student used a volume ratio of 70 to 30 to 1 of ethanol to water to acid. Tick one box that shows the most appropriate volumes she would use to make up 100 centimeter cubes of extraction solvent E. So we've got some important information there, which I've highlighted for you. You should always use a highlighter or circle or underline with your pen in an exam. So let's go through each of these and see which one is the most appropriate answer. Of course, you should pause the video and have a go. So I'm looking at 70, 30 to 1. So going to this third one, and we can see there's 70 centimeters to 30 centimeters to 1. Now it's got the perfect ratio, 70 to 30 to 1. Got an issue. It equals 101 centimeters, and our answer is supposed to equal 100 centimeters. So it can't be that one. And then we look at the bottom one, it's 70.7 to 30.3 to 1. That's 102 centimeters. If you add it all up, that can't be our answer either. We need it to add up to 100 centimeters. And then we go to this top one, it's 63.6 to 27.3 to 9.1. And that sounds good in terms of adding up to 100, but the ratio is wrong, because if you simplify this ratio, we've got 6.9. Yeah, it's not in the right ratio at all, is it? So that's no good. What 6.9 to 3 to 1? We want 70 to 30 to 1. So the only one that adds up to 100 centimeter cubed and is near enough 70 to 30 to 1 is going to be the second answer, which is 69.3 to 29.7 to 1. So it's almost 70 to 30 to 1. Right, let's move on. Figure 5 shows the percentage of rat cells undergoing DNA replication. Some cells contained a protein called cyclin D, and some cells did not contain cyclin D. All cells were in the early interphase at time 0. It took less time for 25% of cells with cyclin D to be undergoing DNA replication than for 25% of cells without cyclin D. A lot of complicated information there that can really baffle us. Let's get down to what this question is actually asking. Use figure 5 to calculate the time difference as a percentage decrease. Show you're working. 2 marks. Pause the Okay, so first thing we want to do is realize that it mentions 25%. So we've got percentage on the left hand side here. And if I go between 20 and 30, that's 25%. And we're looking at these two lines. The dotted line is with cycling and the black line without cycling. We want to find the percentage difference in the time um, for these two readings. So if I go to the first one, we now want to draw a line going down. Of course, you need to use a ruler, but I haven't got one. So I'm going to attempt to draw a line here, which is probably not going to work out too good. And down here, that's going to be 11.5 hours, roughly. And then, oh my gosh, I've never seen such a bent line. And then over here, where we look down, this is going to be 17.5 hours. So we're going to go 17.5 minus 11.5 equals 6 hours. So there's a 6 hour time difference at 25%. But what is that as a percentage decrease? So what you need to do is the difference in the time over the original. So we're going to go, or the difference in the time is 6 hours divided by 17.5 times 100, and it's 34.28%. So you're just finding the percentage change in the time. Cool. And of course, it tells us it's a percentage decrease, and that tells you that you should put a minus at the front there. And there we go. Okay, moving on to the next question. This is, you can see the reference, top right, 2020 AQA Biology A-Level Paper 1. And we've got concentration of particulate matter in micrograms per centimeter cubed at the bottom and percentage of dead cells after 24 hours of exposure on the y-axis. Do the data in figure 6 show a linear relationship between the concentration of particulate matter and the percentage of dead cells? Use suitable calculations to justify your answer. We can see that as you increase the concentration of particulate matter, or as the percentage of dead cells increases, so does the concentration of particulate matter. However, it's not linear. Now we need to justify this. First of all, when there's zero particulate matter, the percentage of dead cells is 6%. So when we work out, we need to make sure that we realize that there's 9% when we're looking at the 5 microgram concentration. So that means because it goes up to 15 and 15 minus 6 is 9%, we have a 9% percentage of dead cells with the 5 microgram concentration. So we can do 9% divided by 5, and that tells us that you get 1.8% per microgram centimeter cubed of particulate matter. So I would expect, if it's a linear relationship, that when we move to 100, okay, that has moved up in a linear way. And what we're going to find is when we work out the percentage per microgram at the 100 bar, it's not going to be following the pattern. How do we do that? Well, we go to the top of the 100 bar and we read off from there and we say, right, that's 42. So 42 minus the original 6% means that we've had a 42 minus 6 is 36. We've had a 36% increase in the dead cells when there's 100 micrograms of particulate matter. So how many percent per microgram per centimeter cube? So we've got to do 36 over 100, and we end up with 0.36% per microgram centimeter cubed. So actually, it's gone down. So this is not a linear relationship at all. We had 1.8 and it's gone down to 0.36. So we don't have a linear relationship at all. So the answer here is no. It's not a linear relationship. And here's just 2 of the calculations you could do to show that. Okay, for this next question, scientists investigated stomatal density on leaves of one species of tree Figure 9 shows 3 examples of square fields of view the scientists used to calculate the mean stomatal density. Now I'm going to add up the stomatas in these pictures for you. You might not be able to see them too clearly, but there's 10 in the first box, 11 in the next field of view, and 11 in the final field of view. And then we've got a little key telling us that these are stomata, so we can identify them. What lines show, white lines show the counting field for the stomata edge. The edge of the white square is 250 micrometers. Calculate the mean stomatal density in the 3 fields of view. Nice and easy. Give your answer as a number of stomata per millimeter squared. So first of all, we need to work out the area. And that's going to be simply width times height. So 250 times 250. I'm then going to divide that by 1,000. Why? Because I want to convert micrometers into millimeters. So I'm going to get 0.0625 millimeters squared. And that's my area of one field. Now I need to find out my average amount of stomata that you would get in one field of view, and that's 10 plus 11 plus 11 divided by 3. So the average mean number of stomata per field of view is 10.67. It's the mean stomata in a field of view. So what's the stomatal density? Well, I'm going to need to divide, we think of density as mass over volume, but the amount of stomata divided by the volume. So it's going to be 10.67 divided by 0.0625, and my answer is going to be 170.66 stomata. I'm going to round that up because the 6 is bigger than 5, so I'm going to round that up to 171 stomata per millimeter squared. And there you go. Tune in for more of the maths videos. Let's get to the
Still here? It's over.